on page 1 talks about the nucleus. So we've got to look at the nucleus, and then we're going to move into protein synthesis. So as you look on page 100, first column, let's see what that says. Oh, no. Okay, well, can't get to it. Have to just wait. And so you see there the learning outcomes. Don't forget, those are great questions. Okay. As you come on down in the column, you see about 4, 5, 6 lines down the bold print term deoxyribonucleic acid. And so DNA is its shortened name is. It says there it contains the code, the plans for all the proteins in our body. And we have a lot of proteins in our bodies. So that's a little bit of information. It's really a lot of information. Some people have figured out that it could be the information in the nucleus. The information that's in the nucleus could occupy at least 1,000 books of an encyclopedia set. A lot of information in there. But anyway, that's the plan for our body, and that's why we come out the way we do. We don't have feathers and so forth. We're not birds. We're people. So information is stored in the nucleus, and it's in a microscopic form. So you got DNA in there, and you come down to the next bold print term in that same column, and you see we have RNA, or ribonucleic acid. Now, we want to look. It should give you a skeleton to put some meat on. Actually, centralized. Is down very low, so they're sweat. The water envelope. You've got those pores, and I want you to notice as you look carefully. You can overlook this easily, but you could look carefully. Come down below nuclear pores, and you see the term ribosomes. Now you got a little dose of that back in chapter 2, I believe it was. At the end, we're going to look at that again just to recoup our thoughts about that. You see the little tiny red dots that are on the purple envelope? Those are ribosomes. And you know that ribosomes are involved in the activity of protein synthesis. Okay? Now notice, if you will come back up on the right side of that nucleus, that little image down there, purple ball, you see rough ER. Remember how we talked about the endoplasmic reticulum, tubular system that connects to the nuclear envelope and connects to the cell membrane, is made of a phospholipid bilayer and transports materials through its tube system, just like we have a tube system in our cities for water to come to the house, and you got a tube system that'll take waste from the house and so forth. Notice that these pores, just pointing out a pore there, a nuclear pore, and that's where some of the endoplasmic reticulum is going to be connected. But notice, look at the membrane. It's a double phospholipid bilayer. Now, this is a little tricky, maybe. You've already had some exposure in the lab, so hopefully what I'm going to say today is not. As you look at the nucleus, You're looking on purple, light blue, whatever color you want to call it, is stretched threads with a light microscope as opposed to an electron picture, and you cannot see the threads in the sense of individual threads because they're so thin. Our eyes don't have that kind of resolution power to see such tiny structures so close to each other. A cell is going to replicate itself. Then the stretched out that I would imagine in the lab you looked at onion root mitosis. That's really nice one, lining up on the metaphase plate, central plate. We've talked about the nucleic acid. We need to say something about the, and that's that little dark staining structure. It's not bound by a membrane protein containing genes that don't code for a protein, these don't, but instead specify the nucleotide sequence of an RNA molecule. Okay? Now, you see on page 102, the next thing is protein synthesis, but let's go back to page 60. As a matter of fact, let's go back to 58 for just a second. Close that off. Now, on page 58, we're down at the bottom of the page and we're looking at nucleotides and nucleic acids. So just trying to get you to get your mind back in order in terms of those substances. Nucleic acids are polymers. The monomers of nucleic acids are the nucleotides. So let's look at a nucleotide structure to get it in our heads, because we're going to be dealing with a bunch of nucleotides in a few minutes in terms of protein synthesis. So you see up at the top, it says on the second column of page 58, second column, page 58, the class of organic molecules in the body we'll cover is the nucleotides. Those are the monomers that make up the polymers of DNA and RNA. Refresh yourself on what a nucleotide looks like. Come down to the next section on the second column on page 58. And the first thing you come to in terms of an illustration is the pentose. What does pentose mean? A bunch of you missed that. That's a 5-carbon sugar. Those are the sugar and RNA. We don't break those down and burn them up as energy. We'll take the hexoses like glucose and fructose, and we'll turn those into ATP through the mitochondria. But we don't do that with the pentoses. We reuse the pentoses of broccoli. We reuse the pentoses of hamburger, which is beef. Okay? And you can go on and on and on. So we don't burn those up for energy. But there's your pentose. And you remember you only got two choices, the deoxyribose and the ribose. So each nucleotide has a pentose. If it's got deoxyribose, if it's got ribose, there's a little hexagon up there. That's just a presentation, a little representation of a nitrogenous base. That's all it is. Hexagon up there. So you can see it's a nitrogenous base. Come down the bottom, there are your nitrogenous bases. 2 purines, adenine and guanine, 3 pyrimidines. cytosine, purines. So remember cut. Cytosine, you're to the more letters required for the pyrimidines. So as a nitrogen, so each pentose group, 3 structures that make up a nucleotide. 60, since we're going to be moving into protein synthesis. Again, look at the middle of page 60. You see those images. The image on your left is D. As far as we're concerned, as far as your pets are concerned, you got birds, cats, dogs, and all that stuff. Always double, maybe in a couple of semesters. But anyway, a ladder. Most of you have climbed a ladder to some degree. And so the rails of the ladder are made of those, the curves there that you see, those purple curves. What are they made of? Look at the top left. And you see 2 nucleotides hooked, bonded to each other. Each nucleotide has that yellow phosphate, has a base, and has a nitrogenous base. Now, they don't tell you what you've got there in terms of DNA. But remember, if you got DNA, that pentose, if it's RNA, you're going to have the sugar. Okay. Now, you come back to that double helix, and you look in the middle, and you see all these colorful little blocks, and they've got T's and A's and C's and G's. Those are your nitrogenous bases that bond to form the steps of the ladder. And notice what they say holds them together. Hydrogen bonds. Remember that all the way back in chapter 2, wasn't it? When we talked about polar covalent bonds. Get that stuff 60. And you see this single strand. Substances compose it. You could look over here to that little picture left on single-stranded RNA. RNA, think double helix. When you think about RNA, think about single strands of nucleotides. Now we're going to go all the way back over to page 102. Page 102, and we're looking at the second column. It says Module 3.7, Protein Synthesis. I'm going to do a little searching today, see if I can find a nice video that I can tuck into week 3 so you can see some of that in action. That can help you understand what's happening. So, you already know the answer to this question. What are the 2 events in protein synthesis? If you know it, type it in. Those of you who are watching. The 2 events that take place in protein synthesis. Okay, you've had a second or 2 to think about it. That was on the last test. Okay. Some of you got it, some of you didn't. Got to get in. Got to be an athlete about this stuff. You got to do it every day. Teach the filing cabinet. Teach the closet door. If you can do that and not look at your notes, you're going to answer the questions and you're going to do well on the tests. It'll get cemented up here, but you got to do it over and over again. I have to do it more now since I'm getting to be ancient. Anyway, come down to the bottom of that column under protein synthesis. And you see, it looks like a little chemical equation, doesn't it? Got an arrow going to the right. DNA, that's the information. And we transcribe, which means to, what would you say? How would you define to transcribe? It means to copy that information. We do it sometimes with a nucleotides, several. You've probably heard of that. It carries the message from DNA. DNA is the repository of that information. And we take the messenger RNA and then we translate it into a protein. So those are the two events, transcription and then translation. So having said that, and you can read about the first 4 or 5 lines right above that, what looks like a little chemical equation, DNA, arrow to the right to messenger RNA, and then arrow to the right protein. And you see it says that the first line up there on that column says DNA is composed of long chains of about thousands and millions of nucleotides. And they're arranged not randomly, but they're arranged in a particular order that guides the development of our body. That's why your arms always come out of your shoulders. They don't come out of the bottom of your feet. Everything's in order. Now, you could mess that up. One of the things that's scary is that people want to, some scientists want to monkey with the DNA so they can make a superhuman. We got people who can't even stay on the right side of the road on Saturday night because they've made choices, and we want them to monkey with stuff like that. I tell you, I have a lot of reluctance in thinking along those lines. But that's what they want to do. That's kind of what Hitler wanted to do. He didn't know what we know at this point. Now, as you come down to that first paragraph on page 103, first column, you come down to a bold print term, it says triplets. Some of you may have triplets. It's a handful, isn't it? Triplet means 3. So as you look at that, it says these triplets each represent an amino acid. Now, we only have 20 or 21 that we use. But we've got triplets for each one of them. Triplets of nucleotides. Now, remember, we identify nucleotides a lot of times with, they all have the same sugar. If you're talking DNA, they got deoxyribose, they got a phosphate group, but they can have different different nitrogenous bases. So look to the right of there, and you see this figure 328. And you see down there, bold print, it says the genetic code. So as you look at that box, do not go memorize it, okay? You don't have to do that. If you were in intermediary metabolism, like I had to take in grad school, then you'd have to learn all that stuff. But you don't have to do that for this particular level course. But you can see up at the top, look at the top left-hand corner of the box. And you come down to PHE, and it means phenylalanine. So those triplets in DNA represent different amino acids. And remember, there are about 20 of them that we use. And so how we mix them, how we put them in and so forth, that's going to dictate a particular protein. Just like you and I put letters in order, right sequence, right number of letters and so forth, and spell something correctly, then we can communicate with each other. We recognize the word by ear, we recognize the word by sight, we understand what's going on up in our head, and then we can apply that knowledge that gets sent to us. Don't go memorize all the amino acids. Don't go memorize all the triplets. A gene that had 1,200 nucleotides and we wanted to think about triplets. You divide 3 into 1,200 and you would have 400 triplets, which means that the protein would consist of 400 amino acids. That's not totally right, but it's right enough to get you through this course. Okay? So, come down to the third paragraph on page 103. And you see it says the genetic code is a list of which amino acid is specified by each possible DNA triplet. And that's that chart over there. Don't go memorize it, okay? Now let's go back to the second paragraph. And you see, during transcription, remember that's copying, each DNA triplet is transcribed, a copy is made of it, into a complementary 3 nucleotide sequence of messenger RNA. So when it makes messenger RNA, which is going to carry the message about the protein, it also consists of triplets. And as you look at the third line in the second paragraph, on messenger RNA, the triplet is called the codon. Okay? Messenger RNA triplets are called codons. Don't go call it a condom. And you remember, A and T always meet with each other, C and G meet with each other. Now we got a little change here, because in the messenger RNA does not use thymine, uracil. So you won't see thymine in a messenger RNA molecule, but we'll see that in just a couple of minutes. Just a couple of things before we move on over to the next thing about transcription. In that third paragraph where you saw the bold print term for genetic code, look at the last sentence. The genetic code is found in all forms of life. Or human. You guys ever get to Earth, blue planet? Just mesmerized with that stuff. But all of them have their own genetic code. And you see it, that code produces a particular shape, produces a particular organism that feeds on particular creatures or plants or something like that. Just amazing variety out there that we're not even aware of. It's like they show you on that show and tell you, had no idea that this thing lived down here at whatever it was, 6,000 feet below the sea, the surface of the sea. Come on down to, let's see, that was a third paragraph. Come down to the next one, in first column. It says, now that we have a better understanding of how DNA codes for a series of nucleotide triplets. There's your definition for a gene. A series of nucleotide, triplets. And each triplet specifies a particular amino acid. That again. So now we're going to move over, as you see, to the second column, transcription. Don't forget to look at flashbacks, all right? And look at transcription. Now, transcription means we're going to make a copy of the DNA of a particular gene. Now, I want you to look on page 105. The number's not going to be up at the top right-hand corner. It's going to be at the bottom right-hand corner. So you're on 105, and you're looking at figure 329. At the top, you see DNA, the double helix. Don't worry about transcription factors. We're not going to get into that, or the promoter gene or something like that. It gets the DNA, the double helix, separates. It opens, kind of looks like the teeth coming at you, doesn't it? They have one side of the DNA has the code. We don't necessarily understand all about that, but that's what we can see. And that's science. Sometimes we talk about what we see, but we don't have it all together. But that's part of the fun, is trying to figure it out. Illustration. That's the DNA nucleotides, as you see. And so you have adenine is going to come back. And you see, just starting at the right-hand side, you see that A is going to hook with the T. The T is on DNA. The A is on messenger RNA. You see it's going to hook up there. You see going to the left, you see the green uracil, okay, messenger RNA. It's going to hook with an adenine. And then cytosine is going to hook with a guanine. Guanine is going to hook with the cytosine. And you see the little red ribbon there? What is that? That's the sugar ribose and a phosphate group. That's the rails of a one-sided ladder, which you wouldn't want to use. Now, you see how the on above it, that's a codon. What? An amino acid. Go back up to the second illustration on page 105. And you see where it's opening up. See the little kind of light brown structure around it? Look to the left and you see RNA polymerase. Anytime you see ACE, think enzyme. And for the most part, that's going to be a protein. So this is a protein that is going to produce a polymer. Polymer is a chain of repeating units, isn't it? So we've got a protein here that is going to construct, going to build. Come on down to the third picture and look at the left of that little chain that's being built, and you see mRNA transcript. That's a polymer, messenger RNA. It is carrying the codes for the triplets. So messenger RNA carries the message from the DNA, and you get down. So if the gene is 400 nucleotides long, messenger RNA is going to be 400 nucleotides, 400 triplets. 400 triplets. If the DNA is 400 triplets, then the mRNA is going to be 400 codons, codes for amino acids. Specific amino acid, right number of amino acids, and in the right order. Just so now we've got the messenger RNA transcript. I want you to go to page 106. Got about another 10 minutes, then we're going to be out of here. Translation. And you think of translation of, since we have a lot of Spanish people coming in, and we got people from India coming east over and close to China. I can't remember it right now. Myanmar, and they spoke a different language. So we got all these languages on the planet. Well, we have to translate so we can all work together. So translation, you see it come down in this first column, come down to the third line. This information is still encoded in the language of nucleotides, and our cells need to then translate it into the language of amino acids so that we can have a protein that'll do what needs to be done. That process is called translation. That is carried out by a third type of RNA. You got ribosomal RNA, which we're going to look at in just a second. We got messenger RNA, and we got transfer RNA, because some people think of it as translational RNA. That's all right, but it transfers amino acids to the messenger RNA. Or that's what it does, transfers amino acids. So you look to the right, you see this little cross-shaped structure. The hydrogen bonds in there, holding that structure in that particular shape. Look at the top, and you see a little football-looking structure with PHE, phenylalanine. That's the part of right down to the end there, and you see AAG in this case, and you see that's the anticodon. The anticodon will match with a codon. And on the other end, it's carrying an amino acid. The anticodon docks keeps the order of amino acids in the order that they need to be. Now they've drawn the little transfer RNA differently to the right, just for the explanation. But that's what a tRNA looks like. It looks kind of like a cross. But then you got this thing looks like a plunger that you break up onions with, doesn't it? You know, it got those little things you can chop up onions or whatever, peppers. Come down to the bottom. Here's how translation works. It's got a P on it. Don't worry about the letter right now. You see the two sections of a ribosome, the large unit and small unit. And you see the messenger RNA lying in between the large unit and the small unit. They're all three of your types of RNA. Messenger RNA, ribosomal RNA, and then transfer RNA. Look down there at the messenger RNA. You see all the little UAC, and it's going to combine with AUG. Okay, there's where your anticodon lines up with your codon. Go to the next step. Number 2. Now you don't have to know the steps. Just look at what's happening in this case. You see it lines up next to the other transfer RNA. And then you see what happens in the third step. The methionine hooks to the glycine. Peptide bond. Peptide bond. And you keep going. You look over to step number, they got 2B there. Here's 2 over on the next page, 2C. And you see this transfer RNA with a little arrow. It's leaving. It's already done its job. And here comes a third one that lines in on the side of the middle one with the anticodon locking into the codon. And then there's the junction of glycine and alanine. Peptide bond. Peptide bond. This goes on and on and on. And so the messenger RNA has the ribosome moving down the messenger RNA. And pretty soon you got what you have over to the right. You have a chain of amino acids. Now, it's not a protein in the functional sense of the word. It's a, you tell me. It's a polypeptide. Well, we mentioned that in the last lecture. Do various things, maybe hook another peptide chain, and we end up with a functional protein. Okay, well, we've been busy here for 49 minutes. I'm going to sign off, but that's what you want to know. You want to be able to explain that. Mr. Pritchett, if you can explain it, you understand it. And that's not all that happens, but that's enough for you. Okay, I'm going to sign off and get ready to go into. See you guys later.